Swayam Prabha. Digital India. Educated India.
Today I am going to discuss medium characteristics and the biochemical pathways. As you know that media plays very important role in the fermentation industries, and I have taken a specific example here, the citric acid production by submerged fermentation from cane molasses. And if you look at here, the cane molasses usually procured from the sugar manufacturing companies, because molasses is the byproduct of the sugar production unit, and this comes to the factory, and this usually kept in the molasses storage tank, and which is located close to the main gate. And molasses measuring tank, this is usually located in the inside the fermentation plant, and then this is mixed with water. The reason is that cane molasses contains about the 50% of sugar, so we dilute with water so that it will be a suitable for the growth of the organism. And then we mix with desirable nutrient and water, and then we mix it thoroughly and pass through the sterilization unit, and then it comes to the we have inoculum fermenter, and another is production fermenter. So the media that is used for inoculum fermenter and production fermenter is usually different, because the purpose of the media in the inoculum vessel is to produce the cell mass, and the purpose to the use of media in the production fermenter to get the desired product. So naturally that the requirement is different. Since the requirement is different, the composition of the media also will be different. So today I am going to discuss this in details. Now if you look at the media characterization, water plays very important role, because question comes of what should be the quality of water. Obviously it should be free from the contaminants, because we should, as the microbial load should be as low as possible, so that sterilization process will be little bit easier. But at the same time, question comes whether the water should be the tap water or the soft water, because tap water will be cheaper, but if you consider the soft water, it will be little bit costlier. So now we shall have, when we use the tap water, we usually determine the characteristics of the water. Characteristics of the water is determined on the basis of the mineral contents of the particular water. If the those minerals doesn't have any influence on the fermentation process, then we can use directly the tap water for the fermentation purpose. Next is your carbon source, which is very important, because carbon source contribute for three different purposes. You see that one is for the growth, another for the product, and it also provides the energy, because it purpose three different purposes. That one is growth, because the cell mass which is very much required, and product which you are looking for run this process, and another is that it gives the energy to the organism for grow and multiply. And nitrogen source is essential for the growth of the cells, and minerals that usually give the balance of the osmotic balance of the media, because you know that osmotic pressure also plays very important role. And also it helps to develop some kind of buffering capacity of the media, because most of the biological system, when the organism undergo the fermentation process, is produce the organic acid, and if this acid formation is there, then pH will go down, and this will affect the fermentation process. The buffering capacity of the media, that plays very important role. And also that this metal ion also has some role to play to find out acts as the cofactor for the different enzymes, that is the act in the metabolic pathway of different microorganisms. Now, vitamins, that is usually acts as a cofactor for the enzymes and are needed for the synthesis of certain product. And oxygen, it should be required adequate amount. And here I want to point out that we all human beings, we take oxygen from the air, but microorganisms, when they grow and multiply, they use the oxygen which is dissolved in the fermentation media. So naturally the dissolved oxygen concentration plays a really important role, because we know the oxygen is sparingly soluble in water. So if we don't supply the oxygen sufficiently, that your growth of the organism will be hampered. If the growth of the organism is hampered, then our whole fermentation process will be affected. Now next is that the addition of precursor, because I can give a very simple example of penicillin fermentation process. If you look at the penicillin G and penicillin V usually used for the capsule, and penicillin G as the injection fluid, and we use for the production of penicillin G, we use the phenylacetic acid, and penicillin V we use the phenoxyacetic acid. And also the media should free from the toxic compound, which is very important. And I already mentioned that you should have the excellent buffering capacity. And another very important aspect is the foaming formation, to avoid the foam, because in the fermentation industry we require the agitator, and in the agitator at the top we have the mechanical seal, and mechanical seal that actually protect the entrance of the air from outside, because if any air enter into the system, this will make the contamination of the system, whole fermentation process will be affected. So to avoid, and if we allow the foam formation, then what will happen? Foam will rise in the fermentation broth, and it will enter into the mechanical seal and rupture the mechanical seal. As soon as the mechanical seal is ruptured, then the contamination of the system will take place. So this is undesirable. To safeguard this, we use some kind of anti-foam oil to subside the foam. Then contamination prevention, that is very important, because some cases we change the composition of the media so that the contamination problem in the fermentation media can be avoided. And simply a downstream processing is a very important aspect, because whenever you do the marketing of the product, it should be in the purified form. The question comes how easily you can do that, and appropriate viscosity to ensure the mass transfer. That is, mass transfer plays very important role, particularly that when we talk about the dissolved oxygen concentration, the oxygen transfer, that depends on the viscosity of the particular media. Now the other different quality that we have, that is the easily availability of the raw materials, because whatever raw materials that we use in the media, that should be available, not only available for a particular season, but it should be available throughout the year. Low cost and surplus, that's also very important aspect. Sufficient supply without transportation cost. Transportation cost, we'll see that as minimum as possible. Then it should not contain any impurities which hindered the product recovery and its cost. More impurities, that our production, that recovery cost will be more, so it will directly affect the cost of production. And it should contain most of the compounds required to support the growth of the organism. Example is the lignocellulosic material. Now let me give you some specific example of the carbon sources. We have first, I already discussed that in case of citric acid fermentation process, we use the molasses. Molasses usually marketed in two different forms. One is called cane molasses, another is beet molasses. Cane molasses is available in India, mostly it is available in India and also in Brazil, and beet molasses is mostly available in the western country. And this molasses contain about the 50% of sugar, and sugar is the basic raw materials. And one of the beauty of this biochemical processes is that from sugar we can produce the different type of compounds. So this is a very good raw materials for the different fermentation process. Particularly, it is very good raw material for yeast fermentation process, for the citric acid fermentation process. Then comes the whey. the cheese whey, which is the byproduct of the sugar industry, the byproduct of the milk industry, and that after separating the milk protein and fat, we get whatever liquid we get, particularly in the cheese making industry. We get the after salting process, we get the whey, and this whey can be used as a carbon source. Particularly, it is a very good raw materials for the production of lactic acid. Then starchy raw materials, that is also very important, which is usually produced from the rice processing industry, particularly India, but West Bengal, if you consider, and as well India, that we are our rice is a staple food, and we have lot of starchy materials that we get from the rice processing industry, and also we can get this starchy raw material from potato manufacturing industry. The cellulosic waste, we have lot of cellulosic waste like we have rice straw, wheat straw, that corn cob, and different type of cellulosic waste we have. And so we have the sulfite waste liquor, that is the waste, the effluent from the pulp and paper industry, that can be used as a carbon source. Also, we can malt extract also can be used as a carbon source, though malt extract contains different minerals which also help in the fermentation process. Now, if you look at the nitrogen source, we have corn strip liquor, but then this is used in the, particularly this is used in the penicillin industry. Then ammonium salt, this is used in the Baker's yeast production process, because here I want to mention that in the Baker's yeast fermentation process, the pH of the media is usually acidic, and to maintain the pH of the media acidic, we don't have to add acid separately. Simple, if we use the ammonium sulfate, when you dissolve in the water, your pH will be acidic. That is good enough to maintain the acidity in the media, acidic pH in the media. Then we have the urea, that is used as a good nitrogen source. Peptone is a good nitrogen source. Yeast extract and soya milk, this is the good nitrogen source that is used. And I told you that nowadays we use also some de-oil cake as a nitrogen source, and we find is a very good results. And some cases we use the water hyacinth as a nitrogen source. There also we get good results. Some cases we use the algae as a nitrogen source. We got the very good results in some cases. Now, question comes, what are enzymes? Enzymes are the macromolecules, the biological catalysts which accelerate the rate of chemical reaction occurring in the biological organism. Now, you can see here that activation energy is this is the activation energy. Now, purpose of this enzyme is that reduce the activation energy, so the rate of reaction is faster. And if you look at here, enzyme and substrate, they form enzyme substrate complex, and then it gives the product. Now, after the product formation is over, the enzyme comes in the original form. That is why this is very specific with respect to substrate. This is I can give the example that glucose isomerase enzyme that acts on glucose and convert glucose to fructose. It will not act any other substrate. If we consider the invertage, it acts on sugar and it hydrolyzes the sugar to glucose and fructose. They are very specific as far as substrate is concerned. The enzymes, the main purpose of the enzyme, as I pointed out, that it reduces the activation energy, so the rate of reaction will be faster. Now, enzymes may be of three types: enzymes of medical importance, enzymes of industrial importance, enzyme used in biosensing. Now, if you look at the enzymes used for the medicinal purpose, it's a three different purpose. One is for diagnostic or prognosis of the disease, as for example of creatine kinase used for myocardial infarction. An enzyme therapeutic agent, we know the streptokinase, which helps to relieving the blood clot, particularly for heart disease or cerebral attack. It is the life-saving drug that is used in the pharmaceutical in the pharmacy. It is largely available. And we have enzymes can be used for the diagnostic purpose. As for example, glucose oxidase can be used for quantification of the blood, that glucose present in the blood. Also cholesterol oxidase is used for the detection of cholesterol present in the blood and the quantification. Now enzymes also used for the industrial purpose. It is used for the textile industry, and it is used for the as a biofuel. You know that biodiesel, I can tell you the biodiesel that is produced from different biological sources, and this is nowadays it is operated by different buses, different vehicles we operate. I can tell you a lot of biogas buses are available in the Western country nowadays. In India also some buses are operated with the help of biogas, with the help of biodiesel. Some trains are in operation. Then it is used in the pulp and paper industry. Paper is nothing but it is the kind of pure cellulose, and you know that for this separation we use some kind of enzyme so that you can degrade the lignin type of materials so that the cellulose can be separated from this. Now it is used in the detergent industry, like in the last lecture I was talking about the stain formation in our cloth, and that can be removed with the help of the enzymes like protease and lipase, and the enzymes also used in the food processing industry. I can give the example of rennin that is used in the cheese making industry for the precipitation of proteins. Now the enzymes that can be used as a biosensor is very important, because you know this is in the biosensor we have two units. One is called molecular recognition unit, and another called transducer. The molecular recognition unit is comprises of the enzymes. This is the immobilized enzyme. This recognize the substrate. Suppose I was talking about the glucose oxidase. Now if the glucose oxidase is present here, then in presence of glucose it reacts with glucose and from the glucolactone, and this change is recorded with the help of some kind of electronic device, what we call transducer. And this is mainly of two types. One is the amperometric, another is potentiometric. Amperometric means that we measure the change of current, and potentiometric we measure the voltage. Then we amplify that, and here we record. And these are the different examples we have. The reasons why biosensors are largely in the market, that we can analyze the sample in a very short time, and number of sample can be analyzed also very high. That is why largely this is available in the market. Now if you look at the biochemical pathways, now every microorganism they have the metabolic pathway, and in the metabolic pathway you will find the different steps are involved, and every steps you have one enzymes, and this is it shows that a series of reactions as how a substrate is converted into product. The intermediates involved in the formation of products is called the metabolites, and they are mainly concerned with the exchange of energy. The interconnection of the network pathways not only indicate the where the substrate energy is diverted, but gives the information on thermodynamics and stoichiometry of the product formation. The example of the glycolysis pathway and Krebs cycle, Kelvin cycle, this is the very popular cycle, that is the popular metabolic pathway that is largely operated in the biological system. Particularly glycolysis cycle is available with the most of the organism, because when glucose undergo the glycolytic pathway, it produces the pyruvic acid, and when it pass through the Krebs cycle is converted into the carbon dioxide and water. So now this is the citric acid TCA cycle, we call it TCA cycle. Now here we can see the pyruvate. Pyruvate is converted from the glucose, it converted to pyruvate with the help of glycolytic pathway. Then this pyruvate with the help of the enzymes, it forms the acetyl CoA, and this acetyl CoA and the oxal acetate they go with the help of the condensing enzyme, what does it synthesis, this produce the citric acid. Now after citric acid we have cis aconitic isocitric acid, oxalosuccinic acid, alpha glutaric acid, succinic acid, fumaric acid. This is the TCA cycle. This is how it operates in our system. Now when here I want to point out that how the citric acid formation take place. Here you see that in the TCA cycle, if you allow the TCA cycle in operation, then what will happen? This will not allow the citric acid to accumulate here. It will slowly is converted to other metabolites. So what we basically do during the fermentation process, if we can inhibit the activity of the subsequent enzyme like aconitase enzyme. Here there is an enzyme called aconitase enzyme. Here we have isocitrate dehydrogenase enzyme. If these enzymes, these two enzymes are inhibited, and then and only then citric acid accumulation in the system will take place. Now it has been observed that particularly when we use the aspergillus niger, then by using the cane molasses, then it has been observed that if you don't control the pH, automatically it will inhibit these two enzymes, and as the time passes on, this activity of the enzymes is totally inhibited. But again, it depends on the metal ion concentration. We find iron and manganese that plays very important role in the citric acid fermentation process. Now if in case of citric acid fermentation process we remove that iron and manganese, then and only then we will find the this kind of citrate accumulation in the system. But if significant amount of the iron and manganese present in the fermentation broth, then we find that this enzyme will be active and it will be subsequently degraded to the respective metabolites. We will get very less amount of citric acid production. So like this, every organism they follow the different metabolic pathway to get the different type of products. I can give the example of this biochemical pathway, how the ethanol formation take place. I told you the glucose after several steps of operation, it produces the pyruvic acid, and this pyruvic acid when undergo the decarboxylation reaction, it forms the acetaldehyde, and acetaldehyde when it undergo the reduction reaction, it produces the ethanol. So one mole of glucose, this stoichiometry is very important. 1 mole of glucose can produce 2 moles of ethanol, the 2 moles of ethanol. So if you look, in the last class I was discussing about the most of the alcohol producing industry. Initially that old days we have seen that the fermentation industry, they have the alcohol production about 7 to 8%. So 7 to 8% means the glucose that or sugar concentration will be around 14 to 16%. But nowadays, due to the development of some special type of microbial strain, it is possible to increase the alcohol concentration to a great extent, up to 14%, 14 to 15% alcohol. Then we can use the concentration of sugar will be about 30%, which is very high. So same time I told you that as we increase the sugar concentration, then the problem is that some osmotic pressure that also develop in the fermentation media. Due to development of the osmotic pressure, the activity of the organism will be affected, and this is the main constant of this process. So we try to develop the genetically modified organism, some kind of mutant organism, so that we can have the high productivity. And industrially, why it is important? Because as the product concentration increases, our recovery cost will be minimized, and if the recovery cost is minimized, then our cost of production also affect to a great extent. Now when we use different type of raw materials for the fermentation process, we take into account, I told you that not only for one particular carbon source, but also we should look into the different 2-3 different type of carbon source, so that the availability of the raw materials that will be more in this market, and we don't have any kind of scarcity of the raw materials to get the desired product. Now I was talking about that different fermentation process like citric acid, penicillin, and when you really we talk about this fermentation process, we have seen that their media composition, they are really different. As for example, I told you that in case of citric acid fermentation process, we don't require any precursor, because precursor some cases is required. Without the precursor we cannot get the desired amount of product formation. But if you look at this penicillin fermentation process, we require the precursor. Without precursor we cannot get the penicillin. So what I want to point out that that composition of the media plays very important role in the fermentation process. And I can give a very typical example to the beer making industry. In the beer making industry, that one important steps is the mashing process. In the mashing process, the hydrolytic enzymes like amylases and proteases, they act on the starchy or protein and to degrade it, so to solubilize this insoluble material. So we find particularly alpha amylase. If the calcium ion is present, then thermal stability of the enzyme will increases. Usually the saccharification that usually prefer at a high temperature, because the gelatinization is a very important steps for the saccharification process. So that is why that if the water contains some calcium ion, obviously it will favor the mashing process. So that the water composition, as I mentioned, that whatever we use in the fermentation process, it may vary from process to process. One industry, the composition will be different as compared to other industry. Thank you.